Good evening, ladies and gentlemen. My name's Professor Pam Shaw. I'm the Professor of Neurology at the University here in Sheffield. And it's my great pleasure this evening to welcome you all to our University of Sheffield Medical School and to invite Professor Winston Hide to deliver his inaugural lecture in a moment. So as many of you will know, Professor Hide moved to our university in September of last year to take up his chair of computational biology and bioinformatics, and he'll explain what all that means in a minute in his lecture. This is a joint appointment between the Department of Neuroscience and the Department of Computer Science. We were very proud because he came to Sheffield from the best university in the world, Harvard University, where he was Professor of Computational Biology and Director of the Bioinformatics Core at the Harvard School of Public Health. I suspect, although I don't know for sure, that Win is a bit of an Anglophile. And perhaps that was to our advantage in winkling him out of Harvard. So he went to school in the UK when his father worked in a position in London, and his first degree was in zoology at the University of Wales in Cardiff. He then went off to Temple University and did his PhD there, following which he spent several years in the deep south of the USA in Texas at the Baylor College of Medicine and the University of Houston. He then went back to his family roots, which are in South Africa, where he built and led the South African National Bioinformatics Institute for 12 years. And then he moved to Harvard, where he was, and I think you were in Harvard for 7 or 8 years, Win. He has a CV to die for, so he publishes his research usually in the highest ranking scientific journals, many publications in Nature, Science, Nature Genetics, The Lancet. He's received many honors and awards during his career to date, including the Outstanding Achievement Award from the International Society for Computational Biology and the President's Award from the South African Foundation for Research Development. Win is an energetic galvanizer of action and a builder of big visions. He created the National Institute for Bioinformatics in South Africa, and since he arrived in Sheffield, he's been a whirlwind of energy. He's already won a competitive bid to provide NHS Genome England with the UK's first dedicated master's course in genomic medicine. This award's only made to top genomics centres of excellence in the UK. He's also well on his way to developing a centre for genome translation in CITRAN, the Sheffield Institute for Translational Neuroscience, with likely support from a major pharmaceutical company. He's a world-leading expert in computational biomedicine, has a track record of successfully catalyzing change, causing a paradigm shift in the way people think about things, and a real talent for bringing people together and creating industry-standard technology for understanding diseases. Already in Sheffield, he's building a new team of world-class experts which can model the diseases we and our families all suffer from in a new way, using a powerful combination of the human genome sequence, drug targets, animal models, and most importantly, clinical information from patients with medical diseases. So together with his collaborations at the Harvard Stem Cell Institute, with industrial collaborators, the Genome England project, and the NHS, he plans to take us to a better place in terms of predicting how our medical conditions will behave and revealing the best treatment approaches for individual patients with conditions. So we're really pleased to have attracted somebody of the caliber of Win to Sheffield. From the first moment that I spoke to him on the phone from Boston, I had the feeling of a kindred spirit. and I felt he would be a wonderful catch for Sheffield, and also a wonderful colleague who will be able to do great things for our university. With his broad experience in bioinformatics and computational biology, together with his understanding of biology. So it's rare to have a computational wizard that also understands the biology of how cells and neurons work, and he has both skills. He's going to really help us understand the complex causes of neurodegenerative diseases that we work on in Citran, and I've no doubt that the knowledge that will emerge from careful analysis of all the big data sets that we get is going to help develop better therapies for patients, help us take a more personalized medicine approach to individual patients, because individuals with a certain disease don't always behave the same way, and also educate our doctors of the future, because doctors of the future are going to need to understand all this genomics. So we're delighted to have you working with us in Sheffield, Win. We're very much looking forward to your inaugural lecture, where you're going to explain to us how you've gone about opening the Pandora's box of the human genome code. It's also delightful to welcome Win's wonderful family to Sheffield. So his wife Tanya is working within the university as well, and his two boys, Alex and Gordon. We're very pleased to welcome you to Sheffield, and we realize what a big move it is from Boston back to the UK, but we're delighted to have you here as a family. So I will say no more other than to welcome Win to the podium, and we look forward to hearing your lecture, Win. Thank you.
Today is an interesting day because I'm going to talk to you about something which is so much older than anything we have in life that we can understand. And actually, it's really old. It's just so old. And today we're going to talk about the human genome, which is part of all the pangenomes that exist. And frankly, we don't represent the genome in the way that it probably approves of. The genome, we are a package for the genome. And it's something which has been replicating itself for many, 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 many millennia, for billions of years. So the genome is an ancient molecule, and it replicates itself. Right, it's working, that's good. And what's interesting about that is it's the only molecule that replicates itself. So it's kind of cool. And we spend a lot of time looking outside the Earth at the comets and sorts of things because we want to find other molecules that replicate themselves. Not quite sure why, but it's always something to do. So DNA has a structure, and we kind of see pictures of it all the time, mostly on television. And it's usually the wrong way, and this is probably the wrong way too. But what's important for you to understand is that it has a backbone. And that backbone pairs with another backbone, and it replicates based upon the fact it falls apart and comes together. It's exactly like a marriage. Sorry, my wife's sitting good. I forgot you were there. And like all good marriages, it has a base, and the bases have names, and the bases may be named after children, but in this circumstance they have names which are named after residues, which are called nucleosides, and they're thymine, adenine, guanine, and cytosine, fondly known as A, C, T, and G. Sometimes we say a bit more than that. In fact, it's quite easy. It rolls off the tongue, C, A, T, T, C, G. I find myself humming to myself quite often, what is the sequence of a particular gene, converting it in my mind. I'm not that sick, but I wish I could do it. So it's interesting because you can make things out of those letters, and the combination of those letters can make more things because the things they make can make things as well. So DNA is a signature molecule for complexity. DNA sits in your nucleus, and it's wound up there. But first of all, before going to discuss where it's wound up, let's take a look at the idea that it contains genes. And genes are actually the most information-dense part of the genome, and they contain very highly encoded information. And there's a start and a stop to a gene, but nobody's quite sure where that is. So people say things like, oh, it's on a gene shore, or I'm on a gene island, which sounds a bit like Daniel Defoe, because it is exactly like Daniel Defoe. No one's quite sure how genes are structured. We know very little about them, but we know more about them than anything else in the genome. So the genes lie interspersed between large segments of DNA for which we have very little understanding of what it does. So we're getting an idea now. Remember, I'm being honest with you. This is not what you normally say in a grant proposal where you pontificate about what you do understand. Here, I just tell you, frankly, we've all got jobs in science because there's plenty to understand. DNA has to be compacted so that it can fit inside the chromosome. And that's because chromosomes themselves are made up of compacted DNA, and the chromosomes themselves compact with each other. And there are pairs of them. And one of the pairs comes from, let's just get if I got this right, the pairs of them come from parents, that's right. And they come into your nucleus and they're built there and they stay there and they're very compacted, but they expand when they're required. So it's a very dynamic thing, the genome. And it's three-dimensional, it doesn't sit in two dimensions like this picture. I'm going to need to put my glasses on. So there's a number of chromosome pairs, 23 being the most popular number that we have today for humans. And there's about 3 billion base pairs in the genome. We're not quite sure because the numbers change depending on who you are. Different people have different numbers, varying by a little bit. And we're not quite sure of the number of genes either. So we say about 23,500, but that's a good guess. And we don't really know what they all do. So I hope I'm giving you confidence in genome medicine. It's going to get better. So what's in a letter? Well, the letters that make up the code are very important. And the reason that they're important is because they have a certain order. And if they get out of that order, you get into trouble or nobody cares. And basically what happens is that when you have a change in a letter, that's when things start to get interesting. If there was only one genome, we could sequence it once and just look at it and we'd be very happy and we could all look the same. But there isn't just one genome, there are myriad genomes. And the reason for that is because genomes reproduce. And when they replicate, they create errors. Not very many. Think about all those billions of nucleotides. But for every few 100 million or so nucleotides, one error creeps in. So when you pass on a genome to your child, you give him 30 errors as a gift, or her. And those errors may mean nothing, or they may mean something. But imagine that life has been carrying on for a while, let's say for a few billion years. The accumulation of those substitutions is quite substantial. Those substitutions which occur that give you grief, as my wife used to say to me, no longer exist. And those substitute because you don't exist. And she said that to me too once or twice, but let's get off my wife. And the other thing that you have to understand is that those which are not going to kill you, but which change you in some way and allow you to reproduce, those are the ones which stay. And those ones are the accumulated variants. And we've got about 4 million of those, give or take 100,000 or two. So genome technology is a very interesting thing because we like to sell things. I was trained in Silicon Valley, where you sell things all the time. You sell them mostly in elevators. You stop to talk to people and tell them about what you do as if they're going to buy your company. And here I've noticed people are a little bit more subtle, which I'm getting used to. I'm trying to be well behaved. I have an English accent, but I'm a South African who was trained in the States, so I'm dangerous about that. But I'm unfortunately not as well off as Elon Musk. So what we need to understand is that in genomics technology, there has been an overturn. And the most powerful overturn I can think of that's created as much of a stir was the microscope, because that was the first time you could reliably and systematically look inside a cell and actually draw it and describe it. Now, you can't look inside a genome and draw it and describe it, but you can sequence the damn thing. And that's what's so amazing, because of course people love machines, and if machines can do it for you, then things get exciting. So the paradigm change is that the data that we're producing as a result of being able to look into the genome is amazingly large, and there's lots of it. So it must be really important, great for politicians. So new machines that read the genome have just been coming out in the last 5 years or so. And what's exciting about those machines is that instead of you personally and painstakingly going through the process of loading a machine for a single experiment and going through that experiment and reading the outcome and being a good student and scientist, instead, the machine does that for you, but it does it in parallel. And that means that you generate an enormous amount of data. In fact, the way these machines generate genomes is going out of style. They're so rapid, in fact, that you can now generate a human genome in about an hour. And what's exciting about that is that you can generate a human genome in an hour. What's not so exciting is nobody knows what to do with them quite yet. However, it sounds great, right? But it's a lot like the Sorcerer's Apprentice, except we don't have a sorcerer and we're all apprentices. And that's kind of how we feel when it comes to dealing with this data. So the amount of this data is increasing dramatically. And over the next few years, and these are the next few years here, you see the lines going up, so you can believe me, there must be more. But effectively, we've been looking at about a rate of about 30,000 or 100,000 genomes being generated worldwide last year or so, and that's creeping up. And that's amazing, because when you think about that, we'll be getting into the many millions within the next few years. And then, of course, factors of that. And that's because the number of genomes you can generate per unit time is decreasing. And that, increasing. And that means that for the same amount of money that you spend, you can get more genome for your dollar. And what's exciting about that, of course, is that the amount of genome you can get is many genomes more for the same price. So if you think about a Ferrari, which I do quite often, a Ferrari Spider today costs about $398,000. I know, I've checked. And, and it's a pity it costs so much, because if it was a genome machine and it had become as cheap as it was a genome, and it had become as cheap to generate as a genome had for the same amount of time since the Genome Project, does anybody want to guess how much it would be worth? How much, Gordy? $10? Way too much, 40 cents. Okay, and I can go for that, so I'm hoping that they invest in new Ferrari Spider technology sometime soon. So I want to give you now an idea of the amount of data that we work with. And this is an 8 gigabit slip-in card that you use, and you can put most of what you do for writing one research paper around that amount of data. It's not too much, unless you have a lot of large files. And certainly, if you work for a few weeks on a paper, that's what you might generate. And that's the amount of paper you could generate in the back of that truck if you really worked hard, or if you looked up all the library accessions. And that's about what you can store on an 8 gigabit chip. Now, if you've been working for a while and you're doing one large research project with a team, you might work in the range of storing data on a terabyte drive. And you might replace that drive every year or so, but that drive will keep all your pictures and all your work as a scientist that you probably need. And it would be the same equivalent of chopping down 50,000 trees and printing out the paper that comes from them. So there's a lot of paper that you're saving, but at the same time, the volume of the data that you're generating just doing normal research day to day is quite high. Now, what happens when you go to the next stage, 1,000 of those terabytes? Well, that's a petabyte. And a petabyte is the equivalent of doing 1,500 full human genome sequences. I know, because I had to spend that much money to buy one. And what has happened there is that if you take all the research libraries in the United States and take the paper from all of those, that's what would be the equivalent of printing out a petabyte. So you can see that just doing a now common or garden human genome sequencing project for 1,500 whole genome sequences, which is a lot, costing about $6 million, would fill quite a number of libraries in terms of the amount. And really, we're generating quite a lot. In 2012, we were generating 13 quadrillion DNA bases a year. Or we could pile 5 Jeremy Clarksons 2 miles high. No, if we just did his head, that'd be 1 mile high, right? So, and then 30,000 human genomes in about a year, but 1 million easily by the end of 2016. So it's cool. We're generating genomes. Fine, I've made my point. And also, they take up a lot of data. And when I came to this university, I was a bit scared because we weren't ready to discuss that. But what's exciting is that the whole region now is discussing ways we can address data in new ways. And talking of regions, if you look across the globe, human genome sequencing projects, national human genome sequencing projects, where hundreds, thousands, or tens of thousands, or even millions of people are planned to be sequenced, are occurring worldwide. And in the States, they're planning a 1 million genome sequence project. And here in the UK, it's very exciting because the UK has actually been the vanguard around the world, and we're generating a 100,000 genome project, of which I think 3 have been generated so far. No, I'm kidding, it's probably 20 or so. It's still beginning. What I wanted to show you, though, is at Harvard we have a very sophisticated way of storing data for genome projects, and particularly for shifting it around. And here it is. And as you can see, there are about 2 terabyte drives sitting on a shelf. And that's how we shift our data around, because that's basically the only way you can. It would take about 3 months to shift around this amount of data if you use the standard internet pipes. So that's not really very viable. So we're looking at new technology which is straining current infrastructure, even at some of the more well-infrastructured institutions. So be that as it may, there has to be a reason that we're doing all of this. And there is a reason, I promise you. And I'm going to talk about the reason for a little while, and then I'm going to tell you about some of the problems it's causing us as a society, and then some of the solutions we're hoping to yield, and I hope it all works. So let's talk about variants. Variants are bits of the genome. This doesn't look like a variant, really. What is it? This is a Manhattan plot. And what this is a series of skyscrapers. They're actually plots representing each chromosome, where we put a dot every time we find a position on that chromosome that is varying within a population of people that have some kind of trait. It could be how high they are, it could be that they don't like the taste of cilantro, or it could be that they have a particular neurodegenerative disease. And in this particular disease, a lot of the people that we've sampled have got a variant at a position on chromosome 6. And it's sticking up there as high as possible because a lot of people, maybe 10 or 20% of the population that we've sampled, have got that particular variant. Now what that means is somewhere on the genome on chromosome 6, and we can localize it right down to a single nucleotide, right there amongst millions of other nucleotides is a variant which associates with the disease. And that means that every time we look at somebody who's got that disease, we find that variant, nearly every time, not always. So it hints that the disease, or something to do with the disease, is encoded nearby in the genome. That's about it. That's as far as we've got. Sometimes we can get a bit closer, but that's about as far as we've got. Unless it directly bang smack hits a particular gene at a particular place where we know exactly how that works, which is very rare. And if that's the case, then we can say that variant is responsible for that disease. But right now, all we can say is we've got lots of good hints. But as you can imagine, I've shown you billions of base pairs, millions of genomes, global reach, lots of problems, and this is not something that your average biologist does every day. So we need computers to do the work, okay? And we'll get into what they do in a little while. So the way things are going, we're going to be able to sequence our own genomes, or at least get our own genome data and an analysis of it done. And that means that we're going to have access to information about ourselves that we've never had before. Really interesting new access, and the way that somebody tells us that information is going to give that person a lot of power. So we've got a concern here, because the way you're told by somebody that gives them a lot of power sounds suspiciously like politics and propaganda. And so the empowerment of yourself to learn as much as you can about your own genome is quite important. And unfortunately, we have an internet-based world where we can go there. But I do see a situation where you walk up at a dinner party and you say to somebody, how's your genome? And they tell you something interesting about it. And Pam, I'm dying to hear about yours. Okay, so, because I know it'll be interesting. So, will I get cancer? That's not an unfair question to ask. And in actual fact, it's a question that Angela Jolie asked. And she asked that question because her family had very bad cancer, in particular associated with a particular gene, BRCA1. And it turned out that she had a very bad prognosis with respect to that gene. She has no cancer. But because she had a very bad prognosis with that gene, she decided to have a double mastectomy, have her breasts removed, and spend the rest of her life dealing with that difficult decision. And basically, it's a bit unreal to think, but basically, you may have to make a similar decision if things tell you that you have decisions to make about your future or your life. So it's a very high responsibility that you take when you have your own genome sequence to interpret, or when somebody helps you interpret it. For her and others, of course, we'd know that members of the family are ill, but that doesn't mitigate the result. Of course, there are plenty of companies that would like to help you. And I'm sure you can imagine that they'd like to make a bit of money on the side if they do that as well. And there are companies worldwide. I've got one here in India, and a couple from the States, etc. And Illumina and 23andMe are two big companies. Illumina does a lot of sequencing, and 23andMe is going to help us understand our genomes. What's interesting is you can do that for free, but it might just be a little bit more open sourcey, and basically you can have your genome sequenced and upload it to a website. And here is a website. If anybody wants to use it, I'm more than happy to explain. And the take-home is that you can go ahead and just share your genome with somebody's random server in Iceland. Well, maybe it's New Zealand and it goes to mega upload, I don't know. But the bottom line is that what you do with your genome might be important to you too, particularly if somebody knows your name. So, talking of names, I'd like to talk to you about my family. And we had an interest in doing a bit of genome analysis. Harvard was given a deal to get cheaper sequencing on their genomes, and so we went ahead and did this. Actually, this wasn't a sequencing project, but it was very near it. We were looking at lots of genes at 23andMe, and I took advantage of that because my sister has schizophrenia, and she has it quite badly, and I thought maybe there's something I can learn about helping her to treat her. And I learned nothing, although I did learn that she has brown hair, which is actually very right. Okay, right. So it was quite useful, and that she's female, and wait for it, she was related to me. Okay, so, but you know, early days. So this whole genome sequencing thing has caught the interest of journalists, and why not? It's a great way to write an article. And I'd like to talk to you about two. The first is Kira Peikoff, who works with the New York Times, and she asked a very sort of scientific question. Do over-the-counter tests work? So she went and bought 3 tests, one from 23andMe and one from 2 others, spat in the tube and shipped it off, and after a few days she got access to a website with her results. And what was quite interesting is that the results weren't quite all the same. One of the companies told her that she had a 20% greater chance than would be expected to get psoriasis. Another company told her she'd have a 2% less than expected chance to get psoriasis. So you can tell that this is frontier technology, you can tell that it's new, and you can tell that it's inconsistent. But you can also tell that people are already trying to make money off it. What's really interesting, though, is all the other traits you can discover. For instance, do you have wet earwax or not? Which you might know already, but then the alleles can tell you. Early rising, get fat easily. Well, these things are very interesting. But the point is, that right here, you can see the genotype. You can see the part of the DNA which tells you that particular information with some pinch of salt as to whether it's true or not. And of course, that's the part we're interested in because that's an absolute value. It's a C, it's an A. We can count it, we can see it. And I'm a quantitative biologist. I love to count things. I love to put them in boxes. And you sit at home and stare at those things and generally organize things, although my wife wouldn't agree with me. And the take home from that is, of course, that you need those skills if you're going to work with this scale of data. So taking a look at another journalist, Carol Gadwala, she went ahead and had a whole genome sequenced by Illumina together with 47 other people, and they met in a room over a cocktail party with iPads with their genomes on them to discuss the results. And they had little folders and they went through that information. And the company told these people that 1,600 genes were of interest, and of those, over 1,200 conditions were associated with those genes that were in the room. 65 were likely to be pathologic, and one individual was told that he would die if he took a particular anesthetic under general anesthetic, and his name is Colin Smith, and he's a geneticist from Surrey. So what was intriguing is that there are sort of random associations, and there are viable things coming out of this, but you can hear it's little data points, it's early days yet. We need a more systematic approach if we're going to make anything useful out of this, but at the same time, it's always going to be fun to surf your genome. In 10 years' time, it is likely, it's very likely, that there'll be strong pressure to have every newborn in every developed country at least have their genome sequenced. And that's because a number of the conditions that newborns are maybe genetically associated with can be helped if you diagnose them early. And that's a good reason. But that newborn information is also very valuable and very powerful. And I'd like to give you some other uses of genomes. There's other new uses for genomes. Social networking, for instance. If you and someone else have both had your genome sequenced, you can share information with each other. Okay? And people do that. In fact, I love to look and see how many randomly distant cousins I share a small segment of DNA with, write to them and find out what they've had for supper. And they send me pictures. We have a great time. But basically, it's just a new form of social networking. Well, maybe it is. But what's really interesting is that new research is showing that if you have friends within a large social network, just a normal one, some of them are more likely to have shared DNA with you than you'd expect by chance. In other words, the social networking companies may be writing to you soon and saying you really should make a friend with so-and-so, okay, if they have access to your information. Also, bear in mind that as traits are associated with food preferences, restaurant marketing should change sometime pretty soon. And certainly, the one that's associated with cilantro is easy to find out, and I can see an occasion right now where my smartphone bings when I go past a shop that's selling a particularly tasty cilantro recipe. But as I write on my blog, you really should think before you spit. And that's because people will pick up remnants of your spit, whether you want them to or not. If you want them to, they can, and of course you can pay them to take the genome sequence, or an artist in Brooklyn can pick up a cigarette that you smoked, as has happened here, and do your genome sequence, and then predict what you look like. And this particular artist, whose name is Heather Dewey-Hagborg, did a series of studies at the Smithsonian, and took the information that brown eyes and Eastern European descent and one other feature could be made into her interpretation of a face. She did a pretty good job, and actually she used that same technology, which involved a 3D printer, to make a representation of her own face, and that's pretty good from an artist's perspective, although I think she may have been involved in some of the aspects of molding that. What happens if you take a genome and do a forensic analysis, or do a pseudo-forensic analysis where you want to model a face from a genome sequence? Well, your face is encoded by genes. That's why it's the shape and form it is. It might have something to do with what you eat and all sorts of other things, but the general characters are going to be genetically encoded. And last year there was a seminal paper about this particular technology, and guess what? They were able to use an overlay of three-dimensional modeling and 3D printing and look at a number of features of the genome, including the genetic history of the individuals. In other words, their not racial origins, but their basically geophysical origins. And they were able to take a good look at that information and do some modeling. And this is the result. On the top, you see models of this journalist's face. On the bottom is a journalist from New Scientist. And they hadn't seen her before. Okay? So now that makes you think. Because if you've given your sequence to 23andMe, well, they have your genome. And if you've been asked to consent, can we do research with your genome? Well, I've already said yes. Well, that means that if they want to research on faces, they can identify you. What's more, they can publish a picture of you because nothing's stopping them. All right? So we have to think about this information potentially in new ways. Now, fortunately in UK we have GPs, and they think too, and the first thing they say is can of worms. Don't do it. And the journalist who I've just described, not the picture of the one previous, Kadwala, she actually asked her journalist for consent, just co-sign on her consent to have her genome sequenced. And he came back with, I'd urge you to think very carefully about getting this done. And more importantly, we do not feel as a practice that we can take this on. And I think that's appropriate. But the real problem there is it's going to have to be something that people take on, because very soon we're going to have genome sequences for everyone. So Genomics England has put together a set of training materials for people such as GPs and others so they can learn about the consent. In other words, if you want to give somebody your genome, what do you have to do in order to ask, what can I do? All right? And the consenting for that is important. So here at Sheffield, as Pam mentioned, we've got a newly minted master's degree in genome medicine. And what we're going to do with that is train nurses, GPs about point of care, researchers, trainees and trainers, and help them to interpret and manage this information. We'll learn a lot, I'm sure, ourselves. And what's so exciting about that is we'll be working with people who provide healthcare instead of people who generally research upon healthcare. And I think that's the key difference about genomes. Genomes are definitely not going to be lost in translation. The take-home is that doctors are going to change the way they treat eventually, and what's useful is that they may be able to predict how you're feeling before you walk in. So maybe the NHS will take less than 10 minutes now for a consult, I'm not sure. Okay. So what I'd like to talk about next is the handling of this tsunami of data. How do we manage to handle sharing of these genomes? I was approached together with George Church at Harvard by the people who are behind the X Prize. And as you know, the X Prize sets goals and provides money to individuals to say, let's see if we can develop a new spaceship. Well, one of the things they were interested in is there a new way of sharing genomes? And we spent a very, very fascinating afternoon thinking about new cool ways that we could set up a competition to test people to find out if there's cool ways to share genomes and which would be the best way. Well, we didn't have the idea of what was right because we were preempted by an alliance, which I'm going to tell you about in a little while, that has addressed this problem in some detail. And I'll give you some information about that in a moment. But George was approached because he is the founder of the Personal Genome Project. It's open source for genomes. And in the Personal Genome Project, you can offer to have your genome sequenced, and they'll do it for free. And actually, it's present here in the UK as well. And it represents a big issue because you have to have informed consent. And informed consent will take you about 4 days to go through, because through that informed consent, you really have to understand what you're doing when you're giving away your genome sequence. It's going to go on a website, and potentially, because your genetic and trait data will be made available publicly, your identity may well be revealed. So if you are trying to hide a disease that you think the insurance company might be interested in the States, that's important. Fortunately, in more enlightened cultures where that power is taken away, here you might just be put in a longer queue. I don't know. So, I'm sorry, it's been the NHS a loss this week. So, informed consent is very, very important. But what's also important to know is that when participants in these studies have their genome data sequenced, they're interested in it. They want to know more. They become empowered. And they start to ask embarrassingly important questions of the scientists. They start to ask questions of their doctors, and they start to share information with each other. To me, that sounds great, because that takes away some of the responsibility, and it shares the responsibility in a way where the individual can understand a lot more about himself in the context of other individuals. And that is the way things are going. Whether or not individuals are prepared to share their genome data, there are warehouses already occurring with walls around them currently, where millions of genomes are becoming stored. Millions of tumors have been sequenced and are placed within the Million Cancer Genome Warehouse based in Berkeley in California. And here in the UK, as you know, 100,000 genomes are going to be sequenced, and I very fully expect, a large proportion of the population will be sequenced at some stage. The state of Qatar is sequencing all of the members of their population, etc., etc. The sharing of that is a problem, but internally at least, that information about each of those nucleotides which change is so fundamental because each of them associates with the disease. And as we get more and more genomes, Eric Lander, who is the head of the Broad Institute in Boston, the equivalent of the Sanger Institute here, has been working for a long time on these sorts of problems, and he makes a very viable point. We will eventually, very soon, be able to look up the genes which cause a disease or which are associated with a disease. They'll just be there because millions of genomes will have been sequenced, and millions of people who have that particular disease will have their information sticking around with those particular genes. The result of which will be a complete catalogue, perhaps as soon as 2020. So if you're into investing, think about that for the biotechnology industry. The important point, though, is that it's going to make a fundamental difference to the way we do medical research. And that's what I'm worrying about, and that's what I want to talk about a little bit more in a while. So knowing your genome is really, really important. And if that information is available before even you're born or at birth, that may well affect how you're treated, what you're fed, and what your parents expect you to do. There are a lot of issues around it. We have to think very carefully about it. And what happens if you have a child who has a disease that won't appear until they're 50? What do you do? So to take forward these ideas, a collaboration of 220 institutes, and if I have anything to do with it, it will be 221 institutes, is joining something called the Global Alliance. And the Global Alliance is for a secure exchange of information about clinical and genomic data, responsible sharing, and willingness of sites to share with each other. All the major sites of these global projects I've been describing with each other to you are part of this alliance. And already we're discussing ways in which we can share information about these genomes so that we can do a better job than just randomly assigning whether you have wet wax ears or not. So, consenting is a big part of that, and I've given that information to you. What's really exciting, I think, is the matchmaker exchange, the last point on the slide. And that is because it will accelerate health. If somebody goes in and is diagnosed, or rather it is undiagnosable for a disease, and it's clearly got a genetic origin, but no one knows what it is, and even if their genome is sequenced and not quite sure what association is there, the matchmaker exchange can allow that person's genome to be compared with other genomes to see if similar ways that they behave, or similar traits, similar phenotypes, similar way they look, similar way they get ill, can be found and associated, and that association may well lead to understanding and potentially even treatment for that person. Okay. So now I'm going to go into the last section, which is actually the science that we're doing here. And really, it's about translating stuff. It's about translating genomes to impact health. And really, that means accelerating genome discoveries, taking what's there and really speeding up the interpretation and banging it straight into healthcare as fast as possible. And you may notice that there's a research institute, Citran, sitting right next to the hospital here. It's a very viable model. And the hospital, of course, has lots of research occurring too. So in that translation, the upfront desire is to have a genomic test for some form of genomic profile. So somebody presents with some kind of genome, you can say, look, here's a story we want to tell you about this particular gene. This is the way you might want to think about treatment. And those treatment guidelines would follow quickly. What's really exciting about that is those tests and other ways of looking at the genome will allow us to predict how better to treat a patient. So Terry Pratchett died very recently from Alzheimer's, or a form of Alzheimer's. And what's exciting about the disease is simply that it's causing enough problems with us, it's highly visible enough that it's giving us the opportunity to generate better understanding. What's not exciting, of course, is there's no cure. So my group has been looking together with Rudy Tanzi at Massachusetts General Hospital at 437 families whose whole genomes have been sequenced. And what we've done is discover new variants of Alzheimer's disease genes that may cause the disease. And what's exciting about that, of course, is that gives us some insight into their diagnosis, and it also gives us some insight into how those genes cause our disease. Terry was misdiagnosed in 2004. People thought he had a minor stroke. Now, that's not unsurprising. Not only was there less genome sequence data and less genomic tests at that time, but also it's a difficult disease to diagnose, this kind of Alzheimer's. But what's important for you to know is right there is a gene that currently is being tightly associated with his kind of diagnosis. And so if he managed to live or be born a few years later, he may have been diagnosed and it might have been able to help him a little bit. Certainly, if you know you're predisposed to getting Alzheimer's, there are things you can do to delay its onset. Another very famous and unfortunate individual, physically of course, is Hawkins, and he has a kind of motor neuron disease, which is progressive weakness of many of the muscles in the body. And for that, only a handful of genetic causes are known. And what interests me particularly about that disease is that there are lots of other genetic contributions which are very complex to unpick. And that means it's a tough problem, which means it's going to be exciting and interesting to do. And instead of looking at 1,500 genomes, I'm fortunate, together with Pam here, to have been invited into a project where we're looking at 15,000 genomes. And that's a large enough number for us to find those little changes, the mutations which aren't as prevalent as the 10% handful I mentioned to you. And one of the things that we'll be able to do is discover genes, new genes related to ALS, and identify clues for their possible treatments. So we have to phrase it in that way because we have no idea what will happen. But at least it gives us hope to make some progress. Well, there has been progress. And since the last 5 years, the genome here has been populated with many, many, many known genes associated with disease. And the colors represent different genomes, different diseases, different genes with different diseases, and the lines represent different chromosomes, and as I mentioned, there are 23. And if you look at neurodegenerative diseases, there's quite a few genes known associated with them. Not a lot, but quite a few. And that's really important and exciting. What's not clear is how they work. And what's super not clear, as Gordon tells me sometimes, is that we don't know how they relate to each other. But we do know that genes do not work alone. People do not work alone. Genes are like people. They hang out in bars, they go to parties, they do stuff together, okay? They get drunk together. And some people get drunk in a way which is very unpleasant. Others get very happy. And that depends on their genetic predisposition. So if you think about genes as people that are hanging out and doing stuff together, that's a really neat way of thinking how they interact. And this blob on the top here is the total interactome of all known human genes. And you can see a blob. That's basically all you can see. So it's very tough to understand that. And the blob has some colors, and the colors just represent some genes which are more active than others. Maybe they've all had a drink, these ones are laughing, and these ones are crying, I don't know. But the point is that some genes can be seen to be more active than others when you are examining them using some of these new technologies I've been describing. And that activity can be how the genes are being decoded and how they're sending messages into the cell. And the more messages they're sending, the more active they are. And those genes which are sending lots of messages together are likely to be working together. We can figure that out. So we're using technology that we use in looking at social networks or speech synthesis or physiological modeling, things which are very, very popular and well researched here at Sheffield, and we're applying it to how genes talk to each other. The slide at the bottom here shows a set of genes which are talking to each other a lot. And we use that technology, that systems approach of looking at more than one gene at the same time, in the Center for Genome Translation. And this is the center that I'm hoping to get rolling and flying fast here at Sheffield. And what's important about this center is that it does lots of stuff, so therefore it's likely to do lots of stuff. No? Okay. So I'm translating, sorry. No, you'll see here that we can use these networks of interacting genes as guides. And what they can tell us is that there's a network of interacting genes that might represent the puzzle piece that helps us better understand Alzheimer's. And if we were able to look at that puzzle piece and just that network of interacting genes, then we'd say, well, when they are very active in an old person, that is where the Alzheimer's comes late, or very active in a younger person, which is going under Alzheimer's, there might be different activities in that same interacting set of genes. And therefore, we can say that person is late or early onset Alzheimer's. When you describe how somebody is ill, that's called a phenotype. And you can say, well, let's make a mouse have late or early onset Alzheimer's. That's not so easy. But let's make a cell have late or early onset Alzheimer's by giving it the same kind of disease things which you get tangles and plaques that you get in your brain. By giving the mouse or the cell the same phenotype, we can then go and look in the mouse and find out what's going on under the hood, what genes are interacting there. And if we can find some similarities between these two, or indeed if we can recapitulate a disease phenotype in a model and look at these interacting genes here, we can assume that some of them might be doing the same thing in humans. So we're doing a bit of a leap of faith. We're looking across millions of years of evolution, but what we are able to do is emulate the disease in a dish, in a zebrafish, in a mouse, or in a fly. And that helps us better understand the disease, and it helps us better pick apart what the causal basis is for the disease, particularly if we're getting variant information from genes from humans, which are pointing us towards the right genes to look at all the time. So the coup de grace for this is to find the cause of the disease. That ain't easy, but it's possible to find causal basis or pieces of that. And if you can find that, you can target it. You can go after those particular things and give drugs which will help alleviate the symptoms or even address the cause of the disease. You can predict how well somebody's going to be based upon how bad that causal basis stuff is. And interestingly, if you know the causal basis, you can go and look for drugs which are known to interact with those causal things. When I say causal basis, I mean a particular process in the cell, say too rapidly growing or dying too quickly, all sorts of things like that. And there are pathways of genes interacting with each other that give us that causal basis. And my lab is particularly interested in describing and discovering those pathways. We're interested basically in doing what Linnaeus did many, many generations ago, where he described the layout of the taxonomy of life. Well, we're trying to look at the taxonomy of pathways. So a lot of people have asked me why I chose to come to Sheffield. And for me, it seemed like a natural decision. And it did not have a lot to do with the gate outside my house. It may have a lot more to do with the friendly people in Sharavale Road, here at the butcher's, or at a restaurant, or the wonderful selection of cheeses. I eat, all right? But what's really the reason, because they have that kind of stuff, but it's not as friendly in Boston, is because here, Sheffield was a powerful, obvious opportunity. Powerful because we have superb science at this institution. Powerful because we have fantastic translational medicine. The mindset is focused on translation. And there are very few people and places that have such a tight focus. I think one of the representations that's sitting here is Dame Pam, who really is the epitome of what should be done in terms of translation. And I've got such a strong interest in that area, we had an immediate rapport, scientifically rapport about that. Then there's the poised potential. We have a superb engineering faculty who actually do engineering, and they do it really, and they're very popular apparently with industry, and they do stuff which is useful. And it's not just about this is the next invention which is going to help you. It is actually helping people and doing things which are important. And that engineering skill is transferable. The modeling that is done here in computers is transferable. And that modeling the way that cells behave and the way that they look. Remember, we looked at the faces. The link back to the genome is very, very important for that. And most important of all, the motivated community, from the youngest graduate student to, may I say it, the most senior, or I'm not going to say oldest, but the most senior executive members of this institution, there's the same thread. And that is that everybody wants to work together to deliver really, really good professional science. And I'm so excited about that, because as a result of working with that, I feel valued, because that's what I want to do too. And this institution is the only place I visited when I interviewed where I really felt, you know, I could do something here and I'm valued. And it's very similar how I felt when I first went back to South Africa. And after 8 or 9 years there, I was able to initiate a Pan-African genome project and able to initiate changes in the way our government worked. Unfortunately, not the right changes, as you know, by the government today. So the final thing I want to say, and the most important thing, about Sheffield and why we chose to come here as a family is we visited and we think the people here are great. And there's 2 of these actually in the park at the Botanical Gardens. And it's just wonderful to interact with people here on a daily basis because people look you in the eye, they smile and they talk to you. And only the place I've ever discovered that before is South Africa. So I'd like to thank you all for your time today and to encourage you to be very careful, but very wide-eyed and very, very open and transparent about thinking about what you'll do with your genome, because you will have the opportunity to do something with it. But if you'd like to help us do something with it, let us know. I'm sure we can make a plan. Thank you very much.